The story of Advanced Biodesign was born of a unique expertise developed for more than 20 years on studying metabolism of cancer cells and especially on aldehyde dehydrogenases.
Program of extreme ambition based on the hypothesis that the uncontrolled proliferation of cancer cells is linked to an aberrant and accelerated metabolism that generates biomass and energy, but also highly toxic compounds which cancer cells have to deal with. To survive, cancer cells use aldehyde dehydrogenases to transform and eliminate harmful compounds and escape from an otherwise irreversible damage, leading to cell death. Advanced Biodesign has developed an innovative approach that specifically inhibits the activity of aldehyde dehydrogenases that are associated with tumorigenesis.
DIMATE is the fruit of 2 decades of research to design and select from thousands of substances the most efficient inhibitor.
Our recent research has brought to light new information about DIMATE's mechanism of action. This suicide inhibitor is able to destroy cancer cells targeting both the highly proliferative cells in the tumor mass and the rare fraction of slow-cycling cells, also known as cancer stem cells, and which are considered to be particularly resistant to drugs and the main responsible of tumor relapses. DIMATE interferes with a crucial detoxification mechanism in cancer cells, causing the accumulation of highly reactive compounds. These are toxic aldehydes and reactive oxygen species. These molecules introduce a noxious redox unbalance, and in the case of aldehydes, it can readily cross-link with multiple proteins and DNA, affecting their function. This cascade of reactions triggers a vicious cycle that precipitate invariably into apoptosis, the death of the cancer cell. Our last experimental evidences indicate that DIMATE can prevent the metastatic spread of cancer cells. It has also proved synergic effects with standard chemotherapies, suggesting that the therapeutic responses to certain drugs can be notably improved if combined with DIMATE. In that context, DIMATE has an added benefit. It hasn't shown adverse effects on immune cells. Rather, it could have a positive effect and act as a potent ally of the current immunotherapies. We must highlight that DIMATE doesn't target a specific mutation or a single anabolic pathway, which are strategies often bypassed by cancer cells using alternative reprogramming or bypassed by the tumor intrinsic heterogeneity. DIMATE, by contrast, hits a downstream and critical process in cancer cells, the detoxification of reactive compounds. DIMATE could be considered a potential breakthrough, and importantly, it has the potential to be effective on multiple cancer types. Professor Regis Costello has been working closely with Advanced Biodesign since 2014. Professor Costello faces everyday challenges of the urgent need for new treatments to combat hematological malignancies.
L'avantage du DIMATE pour les données que l'on a, c'est qu'il n'est pratiquement pas toxique sur les cellules hématopoïétiques normales à des concentrations qui permettent d'éradiquer complètement les cellules leucémiques. Le deuxième avantage, c'est que les patients en rechute et les blastes, les cellules leucémiques de patients en rechute, sont probablement encore plus sensibles au DIMATE qu'elles ne le sont dans la première phase d'induction. Un des problèmes de l'hématologie, ce sont les patients en rechute ou réfractaires pour lesquels les chimiothérapies classiques deviennent de moins en moins efficaces, probablement par des dérives clonales que l'on ne voit pas avec le DIMATE, puisque en ciblant un mécanisme fondamental de la physiologie de la cellule tumorale, il n'est pas possible que des cellules échappent à ce mécanisme de protection en fait. Dans les leucémies aiguës myéloïdes, on peut très bien concevoir le DIMATE en première ligne, en association avec des chimiothérapies classiques, et puis évidemment en deuxième ligne ou chez des patients réfractaires. Donc je pense que le DIMATE peut cibler l'ensemble des leucémies aiguës myéloïdes. Nous espérons, dans l'année qui vient, pouvoir commencer un essai chez l'homme dans la leucémie aiguë myéloïde et probablement dans d'autres pathologies tumorales, puisque cette drogue innovante semble cibler de nombreuses cellules tumorales, hématologiques ou non. Nous sommes très confiants pour le futur du DIMATE parce que les données que nous avons obtenues in vitro, que nous avons accumulées dans le laboratoire et avec des laboratoires en collaboration, montrent une grande efficacité antitumorale, une faible toxicité sur les cellules normales et probablement un spectre d'action très étendu qui permettra de traiter de nombreux cancers.
In 2018, the Boston Consulting Group conducted an in-depth assessment of the DIMATE product profile and has developed different strategic scenarios and perspective on sales potential and valuation.
As part of this assessment, we interviewed and surveyed a range of US and European-based clinicians to understand their view on the potential of DIMATE. First, they're really excited about the novel mechanism of action DIMATE brings. It's a mechanism of action they haven't seen so far, and they're not aware of being developed currently in the preclinic or clinical stage. So it really could mean that DIMATE has first-in-class potential. Further, the planned biomarker stratification approach is something which they consider state-of-the-art. It would allow them to identify patients which would benefit the most from DIMATE while optimally using scarce healthcare resources. Like all products in development, also in oncology, there's an inherent risk of failure during this clinical development. Therefore, it will be important to invest into a clinical development plan which stage-wise de-risks DIMATE and makes it more likely that it reaches the market. If DIMATE reaches the market, DIMATE has true blockbuster potential of sales more than $1 billion at peak sales levels, depending that all envisioned product features are met.
In terms of clinical development, different strategies have been evaluated to maximize the benefits that DIMATE could bring to patients. The objective of clinical phases 1 and 2A is to generate safety data and to obtain positive therapeutic signals. Starting from 2020, this security phase should allow to establish the scope of the therapeutic potential of DIMATE and start negotiating strategic alliances with Big Pharma.
With regard to the overall development plan, Advanced by Design is considering a strategy focused on the indication corresponding to its pharmacological activity profile, acute myeloid leukemia in hematology, and lung cancer or triple-negative breast cancer in solid tumors. In this scenario, the strategy is to quickly obtain efficacy proof of concepts in phase 2B. Let me conclude by expressing my absolute faith for the great potential success of DIMATE. We are convinced as a team in our capacity to identify the critical drivers for an accelerated development plan and the best future funding options for the company. We will need continuous support throughout our clinical phases in order to cure the highest number of cancer. Thank you to all my team and thank you all for supporting us.